in Northern Ireland and went on to receive additional training, including a Master's of Science in Biochemistry, a Ph.D. in Biochemistry, as well as a D.Sc at the Queen's University of Belfast, Northern Ireland. After all his training in Ireland, our paths overlapped at the National Cancer Institute. We were both fellows in Mark Littman's group. I think probably at the time there were maybe 10, 15 postdocs. I want to give Bob credit for inspiring me at that point in time, a physician, in the sense that the disease I was treating, cancer, I really just had a terrible understanding of the basic biology of the disease. So one of the things about that group in terms of training, I think it really inspired me to sit side to side, and this was literally like side to side. You had to see what the NCI benches looked like with really bright PhD scientists who've been thinking hard about the biology of the disease for years, training us relatively ignorant clinicians in what we were missing. So with that introduction, I'm happy that Bob came and accepted the directorship of the Hormel Institute. As you know, Hormel is one of our colleagues in Austin, Minnesota, and he is going to be talking about a systems perspective of endocrine resistance in breast cancer. So Bob, welcome to the seminar.
Oh, thanks, Doug. It's great to be here.
And Bob, I have, there you go, okay.
You got it? All right, good. So what I'd like to do is this morning is give you a sort of a systems biology perspective on endocrine resistance. And against my better judgment, I'm probably trying to fit 3 talks into one here. So I might go pretty quickly on some things, and hopefully quickly enough though that I can answer any questions. And certainly since I'm now one of you, you can get me anytime now. So if there's anything that's left unsaid at the end of this, you'll have plenty of time to catch up. And for those of you that don't think about breast cancer, this slide sort of captures a little bit of the sort of natural history from the perspective of an endocrine therapy. You can see from the figure on the top, you can kind of split breast cancer into ER positive and ER negatives. The HER2 splits almost equally, perhaps not quite, but between the 2 groups. And the thing I think that you can perhaps see most clearly from this is that those that have no estrogen receptor, and so the ones that don't have HER2 as well, the ones that are referred to as triple negative, have a particularly poor prognosis initially, with the annual mortality rate being quite high, rising fairly steeply over the first sort of 3 years since diagnosis. But it falls thereafter, slowly, but it does, such that over time, you can see that it actually ends up such that the annual risk of dying for breast cancer for ER negative disease is actually below that of ER positive disease. So the pattern for ER positive disease is a little bit different. It increases a little bit more slowly, and somewhere around about 3 years it hits its maximum, but then it never goes down. And so the 2 lines cross somewhere around about 5 or 6 years out. And the way that we tend to think of estrogen receptor positive disease over that time period is that the recurrences that arise and the deaths that occur early on are due primarily to resistance to the disease, because we treat for at least 5 years. So anyone who dies during that period is generally thought of being resistant to treatment. The ones that's more difficult and is more prevalent are the ones that go into remission and stay silent for 10 or 15 or 20 years and then recur. And we sort of refer to that as emergence from dormancy. What quite dormancy means is a matter of interpretation, and we'll touch on that a little bit as we go forward. The good thing about this, of course, is that since we've had tamoxifen and drugs like tamoxifen that can target the estrogen receptor, we've got very good first-line responses to these treatments, 60 to 80%, with almost a third of those patients going on to respond to second-line treatment, and even a sixth to third-line treatment. So there's something about the persistence of the estrogen receptor that likely explains those second and third-line therapies. And in fact, most recurrences even at third line, remain ER positive. They don't lose the molecular target for the therapy. And that raises another set of rather interesting issues as to what is the role of the estrogen receptor in acquired resistance. As I said, we've had these treatments as tamoxifen, and we've had tamoxifen since almost exactly 50 years. Unfortunately, while we've done pretty well in improving clinical benefit and pushing out disease-free survival, we've not really moved the overall survival curve much in those 50 years. And that's captured here on this slide. There are a couple of meta-analyses that have been done and major reviews that have looked at this. And what's kind of odd is that the overall survival for an aromatase inhibitor, which are the drugs that block the production of estrogen, versus drugs like tamoxifen or fulvestrant that target the estrogen receptor and are competitive antagonists, and in some cases can target the receptor for degradation. There's really, with tamoxifen, there's no overall difference. So in 50 years, we've dropped tamoxifen as the first-line therapy, moved to aromatase inhibitors, and done so in the complete absence of anything other than a progression-free survival benefit. Nothing on overall survival. And there's a hint that if you switch from tamoxifen to an AI or from an AI to tamoxifen, that sequential therapy can give you a modest improvement in overall survival. But that's largely been done retrospectively rather than prospectively. And so still, and that you can see John Robertson's most recent meta-analysis is still showing that there's no improvement for an AI over tamoxifen. We've actually known this for quite some time. In fact, we've known that anti-estrogens are non-inferior to aromatase inhibitors, even with fulvestrant for quite some time. Unfortunately, the first studies with fulvestrant were done with a suboptimal dose. When we moved to 500 milligrams, which is the current dosing, it actually is not non-inferior, it is superior to aromatase inhibitors. There is an additional overall survival benefit. Now, for the first time in 50 years, we're beginning to see that needle move, if only modestly. Oddly enough, though, fulvestrant is still largely relegated to second-line therapy. And there's still a push to push AIs into premenopausal women. This is mostly postmenopausal women, where you have to suppress ovarian function and potentially have additional toxicities, cardiovascular and others, when tamoxifen is just as good. Why? I'm not entirely sure, and maybe some of the clinicians can tell me, because I suspected if this was any other disease, we would have dropped AIs by now and have pushed some of the other agents. There are problems with fulvestrant and its administration. That's not the point of today's talk. We do have drugs that increase overall survival. This is good. Why, though, do so many ER-positive breast cancers remain dormant? Why, when they recur, can you get second and third line responses, but the disease is now almost invariably fatal? And why has it been so difficult? When we have known the molecular target and we've had drugs that work well, why has it been so difficult to substantially increase overall survival rates? I'm going to argue that maybe we need to think differently about the disease. And so that we've taken a systems biology approach, which I'll explain and exemplify as we go along here. And these approaches really start with a framework as to what the system would look like. And the system we're going to define here is the ER-positive breast cancer cell. We're going to look mostly intrinsically, because much of what happens, although it's modified substantially by the tumor microenvironment, is still intrinsic to the cancer cell. The sensing of the signals that could activate a change in cell fate are done and executed almost exclusively intrinsically. And that's true even with an immune response, where the injection of granzyme B will lead to the intrinsic activation of an immunologic cell death. So how do we define the framework? We've chosen to do that by taking a series of cellular functions as modules, rather than defining modules as cell signaling features. We define them as cell function features that are, of course, driven by molecular signaling. But that allows us to constrain the search space and allows us to focus our modeling in a more tractable fashion, if you will. So the primary functions that we're interested in, they're just listed alphabetically here, are autophagy, autophagy, cell death, metabolism, proliferation, and the unfolded protein response. And you'll get a sense as to why those have been selected as we go forward. The overall sort of framework is captured in the cartoon at the bottom, which we developed with John Tyson about 10 years ago with our U54 in systems biology. And these are all input-output models. I'm not going to show you too many mathematical models. If you want to get a sense for what they might look like, you'll have to look at the supplementary information in our Nature Reviews Cancer description of the model. But what basically we're looking at is trying to understand the functions of the cell, how the signaling that's regulated by the estrogen receptor and altered when it's targeted affect the functioning of these features and how they are coordinately integrated to affect ultimately the cell fate outcome. And so the model must have adaptive, reversible, irreversible features. There is clearly redundancy, degeneracy, nonlinearity, and emergence. Emergence being the property that gives the development of a resistance or a multiple resistance phenotype, that sort of property of what appears to be plasticity. I can discuss it better if you're interested. And we have to, of course, capture the dynamic and directional properties of signaling. And of course, we must also accept that there are mutations, and those mutations often induce stable changes in some features of the framework and the signaling. And the issue with mutations is, we've been able to do sequencing for 15 years or longer. It's been where most people have, over the last 15 years, looked to explain a whole number of features of cancer progression, endocrine resistance being amongst them. Certainly not an illogical place to do. We do think of cancer as being a disease of DNA. But in estrogen receptor positive disease, we've had some issues with really understanding what the role of mutations may be. 15 years worth of data. An awful lot have been sequenced. And yet we find that somatic mutations are really relatively uncommon. I mean, if you take common as being present in greater than 15%, which means absent in 85%, you get a pretty short list of somatic mutations. There are, of course, some gene polymorphisms that have been identified, but the same kind of problem exists. Prevalence is low, penetrance even less. Whether any of these are directly relevant or not is unclear, and I'll show you why I say that in a moment. Perhaps the one case where there is a fair amount of clarity is in mutations in Esr1, which is the estrogen receptor alpha, one of the two estrogen receptor genes, the one that is primarily the target for endocrine therapies. So let's look at Esr1 mutations. They're relatively rare in primary tumors. They are relatively rare in recurrence on tamoxifen or fulvestrant. So they're not predictive or prognostic of value. They've probably been oversold in the context of endocrine resistance, in the context of anti-estrogens, based at least on experimental data for pharmacologic reasons that are largely ignored. It doesn't really matter because the responses in the clinic are pretty definitive. Fulvestrant and tamoxifen are effective following an AI failure. And again, as I said, fulvestrant alone has an overall survival benefit, and that can be pushed a little further with ribociclib or abemaciclib, but not all CDK4/6 inhibitors, which is what these two drugs are. Where Esr1 mutations are absolutely clearly involved is with failure on aromatase inhibitors. They're present in about 40% of those cases, but they're not, as I said, predictive or prognostic. No reason why they would be predictive. You've already got fulvestrant and tamoxifen to switch the patients to, so you don't need to know whether they've got the mutations or not to do that. And we're still stuck with, if they're present in 40%, what's explaining the other 60% of AI failures. We can look at other mutations There's a bunch, they're all actionable and have been targeted, and we've got clinical data on them. The one, of course, that's the easiest one to look at is HER2. And for the HER2 positive, ER2 positive tumors, there's no question that trastuzumab is useful, but it's probably primarily through targeting HER2. And targeting HER2 may be useful in improving response to endocrine therapies in some cases also. The rest is a mixed bag. PI3 kinase, AKT-mTOR, all part of the same signaling feature that's captured in that little cartoon at the bottom. And so perhaps it's not surprising that if one doesn't work, none of them work particularly well. There's no compelling evidence for an improvement in overall survival with targeting effectively any of these. There's certainly clinical benefit, wouldn't argue that, but that clinical benefit comes at the cost of significant finance and toxicity to the patients, without a benefit yet in overall survival. Now, to be fair, maybe something will show up yet because some of these studies weren't designed to detect overall survival as a primary endpoint. But the problem we face, and we saw it with AIs, is that even good increases in progression-free survival when targeting these systems doesn't necessarily lead to an improvement in overall survival. And if that's really where we want to go, maybe we need to look and think differently. Another problem is that these mutations don't always increase with treatment or with progression of the disease. That's the case, for example, with the most prevalent in PIK3CA. And you would have expected those to increase dramatically if they were being selected for in a Darwinian selection process. And I'm going to get to that if I have time today when we get to talking about heterogeneity. So if 15% was the cutoff, we'd have a very few potential targets to go after here in a way that could help most women with this disease. I'm going to argue that we probably now have a pretty good sense of the full mutational landscape of ER-positive breast cancer, and the likelihood that we've missed anything that's highly prevalent and likely to have good penetrance is pretty darn small. Why? Well, when you find changes in the transcriptome, most of them don't turn up, many of them don't turn up in the proteome. It's going to be lower for looking in the genome. So true driver mutations as a sole or primary explanation for endocrine resistance is likely to be limited. And then, of course, and we've seen this really nicely in the AKT-PI3 kinase mTOR studies, you get feedback control. And so a mutation is a broken node in a network, and it's therefore no likely to be more important than a gene that's overexpressed or underexpressed, any single gene that's overexpressed or underexpressed. And in many signaling pathways, metabolism is a good example. There's lots of opportunities for feedback regulation. And so what's the challenge? The challenge, I think, is that if you think of it in the context of individual genes or canonical signaling pathways, you're missing 99.9 repeating percent of what's actually happening within a cell. And because whether you measure it or not, everything in the transcriptome and the proteome and the interactome is still occurring. You get 20,000 coding genes, many more RNAs and protein variants, maybe 200,000 proteins, possibly 1 million protein-protein interactions, and you've still got protein, DNA, RNA interactions. You've got 40,000 metabolites that can interact with proteins. And all of these interactions are occurring at the same time, no matter what you measure. And consequently, the interactome that's ultimately defining the outcome in response to an endocrine therapy is massive in its dimensionality. And the properties of data that has that dimensionality are very different from the features that we are used to looking at when we do our studies in our experimental models. And they have properties that affect how systems work, which I don't really have time to go into today. But what it leaves us with is this sort of problem that we're trying to figure out what the system looks like without being able to see it from every perspective. And so what I'm going to do as we go forward quickly is look at the problem of endocrine resistance from multiple different perspectives in a hope of finding how the system is constructed and how it might work, and then ultimately how we might break it. And so, as I said, we've got a framework. So how can we use this framework to help us with a series of guiding questions, begin to map out what an endocrine resistance phenotype really means biologically and clinically. And so are all endocrine therapy failures the same? I described drug resistance with earlier recurrences and resistance that arises late as a consequence of emergence from dormancy, because there's a resistance component to that. As I said, you wouldn't have emergence from dormancy if the cells had been killed by the treatment. And then, we have our modeling framework. So what features of that framework are altered with resistance? When are changes acquired? And perhaps most importantly, what are the coordinated control functions in the framework that allow for an endocrine responsive phenotype? How are these integrated? And how do they, when they operate together, allow the cell to resist the effects of the stress of treatment? And we approach this in multiple different ways. So this is looking at the first question, are all recurrences the same? We took a simple approach. If we could build a molecular classifier prior to treatment that would predict whether a patient would die early or die late, then probably they're different phenotypes. It's arguable, but it's a simple approach. And we were obviously able to do that, as you can see from the Kaplan-Meier plots. Here, the top was the training set. The second is an independent data validation data set. And we have a reasonably good predictor that can robustly separate and predict whether a patient will die early or late with ER-positive breast cancer treated with an endocrine therapy. So they're likely to be different. How are they different? We don't look at the genes that are selected in the classifier because those are selected for their statistical properties and they don't necessarily, therefore, tell you anything about the biology. And there are many, many more genes that are altered. So we take different approaches to look across the same data set. In this case, we're looking at the transcriptome using Metropolis sampling random walk algorithm to attempt to find those features of early versus late recurrences that might begin to explain why they are different. And the little graph on the right shows you the consensus signaling feature. Couple of things to note, there's nothing canonical about it. You will see that the thickness of edges tell you that there are preferred routes that are more often used or more obviously available than others, but there are multiple ways in which a signal can flow through this, which makes it an interesting challenge to map out. But the features of our framework that are overrepresented here tend to be apoptosis, which is part of cell death, and proliferation. And that makes sense, largely because one of the most effective predictors of responsiveness, at least initially in a neoadjuvant setting, when you're given any endocrine therapy, is a drop in proliferation is marked by changes in Ki67. And so cell death and proliferation are the dominant modules from this perspective. And I'm going to show you multiple other perspectives. So let's now take a set of data. This is an adjuvant letrozole study that we have, matched samples from your primary tumors treated with letrozole. We have the pretreatment and at first recurrence. These are small signaling features that we find using our differential dependency network tool. And the only thing I really want to focus on here is that cell death, metabolism, and UPR are the dominant modules from this particular perspective. And if you take a different approach to this same data set and just do a simple gene set enrichment analysis, you actually find pretty much the same features of our framework are differentially regulated and expressed. Now we get some indication of what specific molecular features may be driving those functions, but I'm less interested in focusing on those at the moment than on the fact that these particular modules are changed. Now, what about primaries and recurrences? If we can predict going forward, pre-treatment, what the outcome will be, is that what's actually driving the phenotype? And so when you look, though, at primaries and recurrences matched to the same patients, what we find is that the genes that predict how patients will respond, you can't look back. When you get to the matched recurrences, those patterns have gone. So the genes that separated pre-treatment from recurrences don't separate post-treatment. Treatment likely has remodeled the transcriptome Now, you would not have expected that if this was mutationally driven. You would, if anything, have expected those features to be amplified, not to be diluted, because you would have expected that those mutational structures to have been maintained and selected for. And so that leaves us with the likelihood that, again, mutational patterns are probably not going to define these phenotypes. But what then are the different transcriptome features that we see between diagnosis and recurrence in this data set? And so here, again, this is the neoadjuvant letrozole study where we used the molecular signaling features to look for. And we looked at using ultrasound as a way of measuring the clinical response. And again, what you see from this perspective is that metabolism is the dominant module. So you're beginning to get a sense that by looking at the elephant, with some idea of what we know we're looking for, we're beginning to see some consistent features arising. So when might some of these features be changed? And now I'm going to switch from looking at patient material because I have to start now looking at our cell line and experimental models. And so here we've taken time course data over 72 hours in T47D cells, which are ER positive, treated with estrogen and or fulvestrant, and looked and modeled the time course changes that we see in molecular signaling, focusing on the edges rather than the nodes, the connections rather than the differential expression. Again, using our differential dependency network analysis. But again, what I'd like to focus on, there are actually 3 features that are shown here, but I've highlighted the key points. You see that there are BCL2 family members that are changed, that XBP1, which is part of the UPR. You find multiple components of MAP kinases that are often associated with proliferation or the regulation thereof. There's a little bit of AKT in here also. None of this, again, is canonical signaling. So we find some features are changed very quickly. The next question is, are any of those changes retained? And if they are retained, how might they be retained? And so this is taking cells that are sensitive and selecting them over a long period of time, finding then what makes them consistently and stably phenotypically resistant. And again, you find some of the very same genes that you found are changed within the first 72 hours of treatment. And here, we're finding autophagy UPR and particularly the cell death features. Of course, some genes get upregulated like XBP1, BCL2, and FKB, and some get downregulated. EGR1, which I'll come back to a little later when we talk about metabolism, and IRF1, which is part of the immune cell, like cell death features. And you can take these types of data and begin to build mathematical models that predict the outcome. I'm not going to spend much time on that, but this is just to show you that you can take the sort of data that I've just shown you and build a mathematical model that is predictive of how you can perturb the system excuse me, to get changes in cell death. And the little graph at the bottom right there simply shows you that the mathematical model in simulation is pretty good at predicting what you get when you perturb the system, in this case with the dominant negative IRF1, that can predict the outcome. This is not the whole system by any means. It's a small piece of a small part of it, but it shows you how you can begin to model pieces of this that are both predictive and reasonably accurate. Some of the changes, of course, are functionally relevant. The dominant negative IRF1 study there shows that for IRF1. This shows it for XBP1, where you can overexpress it in sensitive cells and get resistance, knock it down in resistant cells and reverse resistance. And as others have shown, its expression is associated in a univariate sense with clinical outcome to tamoxifen. And this is a component, again, of the UPR. That implies that the UPR is a component of the system we need to understand better. Moving along fairly quickly, one of the issues that we often get asked is, but you're looking in cell lines, how do they refer to look like tumors, and aren't cell lines just totally different and not particularly useful? Well, no, is the answer to that comment, which surprises some people. We actually find exactly the same features in the cell lines as we find in the tumors. They are not represented identically. The features are the same, the modules are the same, the genes that represent them have significant overlap, but they are certainly not identical, nor should they be. You take two tumors and they wouldn't give you an identical readout. And why should cell lines be able to teach us something along the lines and at the level that we're looking? Well, if they couldn't, we wouldn't have learned anything from yeast. We wouldn't have learned anything from worms or Drosophila. And yet our initial understanding of how cells go through the cell cycle was learned from those types of models. So, of course, there is evolutionary parsimony. There must be things that occur in cell lines that occur in tumors. It's just how can you figure out which things are conserved well enough to teach you about the system. And again, when you look at it from this perspective, and this is now looking at the proteome, we find that metabolism and the UPR are the dominant modules in the signaling that's activated from looking at the question from this perspective. The UPR, for those of you who don't remember, is an ancient stress response. You can find it in yeast, for example. In mammalian cells, there are three arms to it. It is regulated upstream by glucose-regulated protein 78, which links the stress response pathway to glucose flux and concentrations within cells. It can be triggered by changes in oxygen, nutrients, a number of different stressors. DNA damage can activate it. And what it is a quality control system that regulates the improperly folded proteins that can refold them or then send them for degradation in response to stress. Energy, of course, is required for protein folding, and so the UPR is linked closely to the production of energy at one end and to the maintenance of cell death and proliferation at the other end. It is simply one module in our network. It is not the only or the most important, but it is relevant. It is upregulated with anti-estrogen resistance. You knock it down in resistant cells, you sensitize them, you overexpress it again, and it gives you resistance. The signaling features are different than canonical signaling. GRP78 is upstream in the UPR module. BCL2 is in the cell death module canonically. And from the canonical UPR, that would be a function of junk and chop signaling. But if you remember, if you look at that little panel in the middle with the red arrow there, that is our prediction of how the signaling would work from our DDN model. It's non-canonical and it implies a regulation by XBP1 of BCL2, and that would be the connection between UPR and cell death. In the context of endocrine resistance. And that's captured a little bit in the Western blot that you see above, that BCL2 and GRP78 track pretty well together. And what this slide shows you, it's a little busy, but basically the endocrine regulation of BCL2 is lost. If you knock down XBP1, BCL2 goes down. Actually, you can also change BCLW because there is degeneracy within the BCL2 family with regard to control of cell death. It's not just all BCL2. And that's really what I'm trying to get at here, is that although we found BCL2, we use that as an indication of where to look, not that BCL2 is the be all and end all of this control and integrated regulation. The bottom three panels go from the left and show you that XBP1 regulates autophagy. That also is a consequence of its regulation of BCL2. And the middle, those two red ellipses show you where BCL2 sits in the integration of apoptosis and autophagy. And most people are familiar with its role in the mitochondria and protecting cells from apoptosis. But it also sequesters Beclin-1 and regulates the ability of cells to activate autophagy. And so BCL2 and its regulation by XBP1 is a coordinated integration piece of how different components of our framework, and how different modules therein are integrated and function together. And there are lots of examples of that as we go forward. Inhibiting autophagy by Beclin-1 siRNA or 3MA or regulating BCL2 also affect responses to endocrine therapies in a way that's similar to upstream regulation of XBP1. Autophagy itself, if you block it with chloroquine, we've shown that this works quite nicely in sensitizing cells to tamoxifen particularly, but also to some degree to fulvestrant. There are a bunch of clinical trials looking at this ongoing. And to keep the theme of understanding the elephant, if you like, we're not going to ask one of the other questions. We've got some ideas that we've got integration of UPR, apoptosis, and autophagy. We've seen that some changes that occurred early are retained. Now let's ask, how are some of those retained? They're not likely retained as a consequence of mutation. They are much more likely a consequence of epigenetic modification. And this is some work that Lena did with us using HDAC and DNMT inhibitors. Now, these are taking hammers to crack walnuts, but nonetheless, they establish the principle that some of the key features of our molecular signaling that likely integrates and executes the cell death outcomes are being now maintained by epigenetic modification. And that links again back to another coordinating and integrating function, which is that epigenesis is basically a metabolic process. The methyl or acetyl groups that are being disrupted by HDAC and DNMT are purely a consequence of the metabolic process. The TCA cycle is a primary source of the acetyl groups, for example, for acetylation, and ATP for methylation. So that, again, begins to integrate changes in what's occurring in cell signaling that's regulating the metabolisms, altering DNA, that's controlling the integrated function of these modules within the system as defined by our original framework, and that reflects all of the early changes that I showed you in the patient material data. So if we're going to look then at metabolism, what are the metabolic adaptations that contribute to resistance? And of course, again, we've got our endocrine resistant matched cell pairs to look at. We've got transcriptome and metabolome data. And so we can begin to map the metabolome onto the transcriptome to get an understanding of how the signaling integration controls features of cell metabolism in a way that addresses the potential cell stress. And if we're activating the UPR as a central cell stress response, we must be changing glucose metabolism and other features of metabolism. And I'll show you that we are in a moment. In fact, you already get a sense of that from this map. And I realize it's a bit busy and difficult to read, so I'm going to focus in on just a couple of features. Here is the glutamate feature that gets at metabolism. We know that GLS, that metabolizes glutamine, is increased in ER-positive tumors. We know that it's affected, it's an effective combination when it's combined with everolimus. There are now clinical trials ongoing with glutaminase inhibitors. They're beginning to get at this feature, and it's only one feature of the overall system. And just to link that into cell survival, you see I've highlighted here where the BCL2 family members and ESR1 feature. ESR1 is not surprisingly at the heart of this. But also you'll see that EGR1 is regulated, and I showed you that earlier. We've subsequently confirmed that it's a key integrator of regulating cellular metabolism and cell survival. And we see other things, close to Doug's heart is insulin and IGF-1 signaling, which is absolutely in here. Now, whether it is the metabolic features of perturbations in IGF and insulin signaling, as opposed to the mitogenic signaling that's important, I can't really tell you, but you can't actually have one without the other, because you can't activate the mitogenic signal request if you don't have the energy to do so. And that may also be how these feature primarily in the context of endocrine resistance, that it's controlling intracellular energy pools because they are also affected differentially with the acquisition of endocrine resistance. So let's then follow the money on energy metabolism. The obvious place to look is glucose. We've known this since Otto Warburg was playing with his tissue pieces in the 1930s and the 1950s. And if you look at this in our experimental models, the panel on the left, it says glucose shows you the LCC9 of the resistant cells. They take up glucose at the same rate as estrogenized LCC1 cells, which are sensitive. You treat LCC1 cells with fulvestrant or tamoxifen, and that uptake of glucose is dramatically changed. Now, if you just take this as a simple input-output model and you say glucose in, ATP out, it's a gross oversimplification, but it is a useful simplification. That's shown then on the right. And you can see on the left bars on that right panel for ATP that tamoxifen and fulvestrant dramatically drop the intracellular concentrations of ATP in sensitive cells, but don't really affect them much in resistant cells. The point, though, and it's an interesting one, is that dotted line. If you look at that, the resistant cells are proliferating at the same rate as estrogenized LCC1 cells, but doing so with the same intracellular ATP concentration as is present in cells that are dying when they're sensitive and it's reduced. So that implies that resistant cells have a more ATP efficient metabolism, and it might create a collateral sensitivity. That's reflected in an upregulation by estrogen of the GLUT1 transporter, which is the primary, but not the only. There is degeneracy within glucose transport. There are multiple transporters. This is the primary one. It's upregulated by estrogen. It's upregulated in resistant cells, and it's upregulated when you transfect cells with XBP1. So upregulation of the UPR can induce upregulation of GLUT1. And clinically, that's related to FDG PET studies that show that changes in glucose metabolism or uptake in tumors is associated with event-free survival in ER-positive breast cancer. So far and throughout, you can link pretty much every piece of what we've seen back to a clinically relevant observation. But of course, it's not just glucose, and glucose is not the only solute transporter that's affected that might allow cells to scavenge as part of their survival mechanism. And we've mapped out the SLCs also. In addition to GLUT1, there are a bunch of others that are changed, including LAT1, which I don't really have time to go into today. But if we follow the glucose metabolism, one of the rate-limiting steps is the uptake of glucose, of course, and the HK enzyme. But one of the integration points, which is where we really want to look, is where are there integration points between glucose and energy metabolism and cell fate. And the PFKFB3 enzyme is one of those coordinating signals because its product is required to complete glycolysis, but its product is also associated, as is shown in the left cartoon, with affecting CDK1 and its ability to phosphorylate P27. And so flux through glycolysis is sensed by and affected by PFKFB3, but it also then links this to the ability of cells to enter from G0 into G1 and into S of the cell cycle. So this is a glycolysis feature that coordinates cell proliferation, and cell proliferation is key, as I showed you in many of our early molecular signaling and with the Q67 data. And if then there is a collateral sensitivity, blocking that enzyme should have a greater effect in resistant cells and in sensitive cells, and that's shown in the right panel. That occurs in MCF7 and multiple different variants, and then it also occurs in T47D variants. And in this case, the cells die very rapidly through a ROS-dependent necroptosis as opposed to apoptosis, but the complexity of the cell death module is a different story. Cell sensing of nutrient energy, I want to get back to this very briefly because it is what's targeted by PI3 kinase AKT-mTOR signaling, but it has multiple degeneracy in its control. The GRP78 can control AMPK. AMPK can also sense changes in ADP, AMP in energy pools. But of course, the UPR can also affect autophagy as a way to get around the energy deficit. So if cells no longer proliferate, they then have all of the excess material that they needed to proliferate. They're not going to use it. And so they can use autophagy. I'm going to run out of time if I'm not going quickly already. And so that leads us to this particular model, which I will explain very briefly and very quickly. And it's not meant to function precisely this way. This is the simplest way to explain it. You give an endocrine therapy and you reduce glucose uptake. Glucose uptake reduces ATP levels over here and can induce autophagy through AMPK, but it also is sensed by GRP78 that activates the unfolded protein response. And if that energy level isn't enough, change isn't enough to activate it, the production of unfolded proteins due to insufficient ATP will activate it. And then you get a multiple control for activation of autophagy. So the cell can recycle what it doesn't need. That can allow cells to survive. You've blocked cell death through the concurrent activation of BCL2, which buys more time. And you've got multiple alterations in cell metabolism as the cell is coordinately attempting to get back to homeostasis. And so that's how this system functions as an integrated intrinsic system. But I want to, in the last couple of minutes, and we can come back to this if anyone has any questions about it, explain why the external environment is critical in affecting this internal environment. And that gets to heterogeneity, because you can't avoid heterogeneity, particularly in breast cancer, which is notoriously cellularly and molecularly heterogeneous. Of course, when you talk about heterogeneity, you have to define what type of heterogeneity. Is it genetic? Is it molecular? Is it cellular? Is it perfusion? Or is it just drug response heterogeneity? No, some of those forms of heterogeneity are related to each other, but the one, of course, that we're most interested in is drug response heterogeneity. But we're sort of constrained because our ability to get prospectus and to look into that heterogeneity, particularly from clinical material, is challenged by our ability to get that material and when we can get it. As you can see, we have a sampling bias problem, which is well known if you're sampling a heterogeneous tumor. We have spatial heterogeneity. Different tumors within the same patient will not look identical. And we have temporal heterogeneity because those change, as I showed you earlier. The molecular profiles change with treatment and time. And we are stuck with our perspectives that look at it with these snapshot data that are confounded by bias in sampling with intertumor, temporal, and spatial heterogeneity. As this review had suggested, one of the few ways to address this is by taking a systems biology approach. So let's look at heterogeneity. I would note that the ER expression and HER2 are heterogeneous, but the drugs still work. So it's not the only story, but it is clearly a critical component of it. So first, the cell lines, much maligned, which I've already told you do have features that you can find in clinical samples, are actually not phenotypically homogeneous. Even in the most sensitive cell lines, you have sensitive cells and you have persister cells. All the cells don't die. And in some cases, you can have resistant cells present in the population. And so if you look at our LCC1 on the left, we for sure know we have sensitive and persister cells. We don't know if we truly have resistant cells. But how then does the estrogen regulate the relationship between sensitive, persistent, and resistant cells? And you can model this. This is a Waddington landscape study looking at some of Dixanton's data, showing the emergence of estrogen independence and hypersensitivity. And I'm just going to cut straight to the chase here. What we find is that the estrogen receptor functions as a bistable switch, that it's reversible whether that's activation by estrogen receptor or by phosphorylation by growth factors, but its preferred state is on, and it's easier to turn on than it is to turn off. And so when we treat every day for 5 years and maintaining the brake on, we are forcing cells to acquire a resistance phenotype, and it's harder to reverse. The other issue is that cells talk to each other, and this is taking sensitive cells and resistant cells, mixing them, treating them, and looking at what cells survive. And in this case, both in vitro on the left and in vivo on the right, we find that resistant cells enable sensitive cells to survive. So the cells are communicating. In fact, what's happening is the R cells are remodeling the S cell proteomes such that the sensitive cells begin to mimic resistant populations. And that is a consequence of both juxtacrine and paracrine cell communication, which will bring Doug back to our days at NIH. This shows you studies we did with Lucifer yellow and M. cherry, one of which Lucifer yellow, but not M. cherry, can go through gap junctions, but they're both packaged into microvesicles. So both are occurring because if you block either gap junctional intracellular communication or microvesicle secretion, you reduce the ability of resistant cells to confer resistance upon S cells. Interestingly, though, you combine those two, block both gap junction and microvesicle secretion, but you get no further addition value, which suggests that they're both communicating similar signals. And this is almost my final slide. Because what that leads us to, and you would have got this sense anyway if you're willing to accept that this is likely primarily not a mutation-driven phenotype other than Esr1 mutations and AI resistance, that this is likely an ecological problem, not an evolutionary problem. And so this is taking these cell mixture data and using ecological models to understand the changes in population dynamics that occur as a consequence of treatment with an endocrine therapy. And basically what we find is that endocrine therapies remodel cell populations dynamically along this mutualism parasitism continuum. Because they're capable of enabling S cells to survive, what is most consistent is that we have an element of mutualism where the sensitive cells and resistant cells talk to each other and enable each other to survive. And we can talk about that separately. But the one thing that I would note, and it's in this panel here, is what we see when you treat a sensitive population with an endocrine therapy is you get a change in the carrying capacity of the population, which means the population can no longer sustain its size at the size that you would have had in the absence of the stressor. And what that implies is that one of the reasons that we get dormancy is because, yes, we constrain the growth of the cells, but even after we take the drugs away, the cells remain incapable of growing to the same size as they would have done before, because the carrying capacity of the population has been rewired as a consequence of their adaptation to resistance. And it's only much later that the cells are capable of escaping that and changing that carrying capacity and allowing the tumors, then the micrometastases, to grow to the point where they were detectable and you have metastatically detectable disease. And so ultimately, is this a consequence of evolution versus ecology? I'm going to argue it's ecology over evolution. Depending on how you do it, you see some Darwinian selection, of course, but you also see mutualism. There's some lovely work that Bob Gatenby in Miami did, who looked at MDR1 resistance. He showed that sensitive cells restrained R cell growth. We don't see that with endocrine resistance. We find the population functions more on a communication that is mutualistically beneficial. But what it means is that rather than studying mutations and using that and evolutionary biology principles to understand the nature of heterogeneity, we likely have to take a much more ecologically based system. And what's good is there's lots of evidence for that in other areas of biology that we can learn from. It also means that if we understand that as the nature of what's driving heterogeneity, it gives us different ways to approach how we can control it in order to get good clinical outcomes, or at least better ones. So I've sort of given you a whirlwind tour at 30,000 feet of an awful lot of data collected by a lot of people over a lot of time. And I need to give you a sort of a bottom line take-home message that I hope will at least have you thinking. I think what hopefully I've been able to convince you is that endocrine resistance reflects the capacity to coordinate multiple activities among and within critical cellular functions. We've chosen to define those functions. We're not close to adding additional ones, but the 5 that I described earlier on make the most sense. We've shown how they're integrated and how they coordinately respond such that the whole system responds as necessary to the stress. The cell takes every opportunity it has to adapt to the stress of an endocrine therapy. Not much it can do if you get an Esr1 mutation that drives the system and certainly stops cells from responding to an aromatase inhibitor. But we already know what to do. We can treat with fulvestrant, with one of the CDK4/6 inhibitors, probably not palbo, but one of the others, and you can get an advantage on overall survival, even in patients that have recurred on an AI. So resistance doesn't require mutations. They occur, they can modify the system, but they're not required. Epigenetic changes probably are required. The cells function by sensing changes in their metabolism, activating a centralized UPR, and integrating autophagy, scavenging, and other functions within the cell to get what they need to restore homeostasis. So they can adaptively rewire. Sometimes that creates collateral and actionable sensitivities. And so we don't need new genes, new mutations. We need to understand how rewiring works. And we need to understand how cells communicate. I'll leave the acknowledgements there. If you were paying attention, the people who did the work were listed at the bottom. Too many people over too long a period of time to pull people out specifically. But I would like to thank the patients who contributed to the clinical studies that took up the first or third of my presentation. And if you remember, those were mortality data. And so those patients gave us of themselves, and they're all dead, and they did it knowing in all likelihood that it may not benefit themselves, but it would benefit others. And it is that wonderful altruism from our patients that allows us to ask and answer questions in ways that otherwise we could not. And I'm going to stop there.
Great, Bob, thanks. There's a little bit of time for questions, so I would ask the audience, if you have a question, put them in the Q&A part, and I'll try to take a look at that. And I think you can probably raise your hand, and we can unmute you too. And Sandy will keep a look at that. Bob, I do want to ask a question that came up. You talk about the various types of heterogeneity, and you might have mentioned this, but how do you model organ-specific heterogeneity? In other words, the cells that end up in bone, liver, lung, brain, obviously find themselves in a different environment. And how are you thinking about that?
You know, it's a great question. I don't have a great answer. You know, I think we are constrained in one sense that we can only look when we know to look. So to get the material to study, we can only do it once those lesions have got to the point where they're clinically detectable and we can assay them or biopsy them and assay them. And that's quite late in the process. Certainly, it tends to be late in the progression of the disease from the patient's perspective. Excuse me. So, you know, there's a time constraint that we have there. You could try and look at, you know, circulating DNA or or CTCs, they probably also tend to arise relatively late. And, while they are almost certainly seed some of these, and some of the biology of looking at those can be helpful, I'm not sure how definitive it is that the one that you found or the ones that you found are the ones that were responsible for what happens later, or for what's happening at the same time somewhere else, because often you've already got metastatic disease by the time you detect those. We're kind of stuck. You can do single cell sequencing and try and look at the transcriptome. Technically, they're more difficult to look at the proteome just because of sensitivity. Modeling it, there are technical issues.
We're at the end of the hour, and I would encourage people to email Bob if there are other questions. I do want to ask one last closing question. So as a clinician, I'm worried that the direction the field is going is to prolong tumor dormancy with things like CDK4/6 inhibitors, as opposed to addressing the issues that you've outlined and trying to kill those dormant cells. And in some ways, I don't want to make them more endocrine sensitive. I just want them dead. So what do you think we should be doing? Should we be pushing the metabolism button harder, or where do you think we should be going as a field in trying to not just, say, okay, 15 years of the CDK4/6 inhibitor will increase overall survival by a smidge, as opposed to completely eliminate the need to give 10 years of endocrine therapy?
Yeah, that's a wonderful question. And, if I had the right answer, I probably wouldn't be sitting here talking to everybody. I'd be heading to Sweden. But, I think there is value in not killing all of the cells if they never come back. Unfortunately, that's not the case with the CDK4/6. You would have a functional cure. You wouldn't have a true cure, but you'd have a functional cure if you kept cells dormant and they never came back. One could live with that. Will we achieve that with the CDK4/6 inhibitors? I suspect not. It's a great question. We've had antimetabolites. They're probably they are one of the oldest class of cytotoxic drugs, and we still use them. So that should tell us that is certainly a useful place to look. I think it ultimately comes down to understanding how to combine what we can combine in a way that gives us the greatest cell kill at the least pain and toxicity to the patients. And I don't think we know how to do that yet. But we can model those extensively in our cell culture and animal models and using mathematical models in a way that you can't do clinically. And so there certainly are approaches that will allow us to reduce the search space and potentially optimize our current therapeutic options in ways that we can't do empirically in patients, but yet that's what we still do.
Okay, I think we better. Carol Lang just asked a question. I'm going to, maybe a clinical question. I said, why do you think Palbo is not so good relative to other CDK4/6 inhibitors? I didn't actually mean to imply that. I think they're all the same. I'm just arguing the fact that suppressing tumor dormancy better is part of a solution, but to me as a clinician, it's not the ideal solution. I'd rather just say, you have a very slowly proliferative tumor. It is endocrine sensitive now. I have a new therapy where I can try to eliminate those cells in a year or keep them dormant forever, and I'm going to do that. So anyway, yeah, they're clinically they're pretty much all the same.
All right. Well, thank you so much, Dr. Clarke, for a great presentation. Please, everybody, reach out to Dr. Clarke if you have more questions. And we'll all see you again next week.
Well, thank you all. Sorry that I went a little over, but I really appreciate your time.
All right. Thank you.